So we're going to start again at number 6 because the first 5 are the same. Now we're going to change these steps. So step 6, remember we had step 1 through 5, we turned on the B cells. Okay. So the helper T is going to activate a B cell. So just like the T cell, I'm going to turn on a B cell. That matches. What do you think it's going to have to match?
Original antigen.
The original antigen, that specific concept. So just like the T cells, I'm only going to turn the B cell that matches the original non-self shape, just like in the T cell system. And what do you think that B cell is going to do once it's got the orders from the general? Pick a guess.
Clone?
It's going to clone. So step number 7. The B cell clones or proliferates. We call those plasma B cells. Can anyone guess where they are if they're plasma B cells?
In the plasma.
In the blood. And they're going to match the original non-self antigen. They're clones. Okay? Great. So, whoop-de-doo. Why am I making B cells? Let's do step 8. This is a unique step here. I'm going to change colors. All right? So, plasma B cells make antibodies. They make antibodies. So T cells kill things. B cells do not kill anything. They make something. What they make are antibodies. So we have to take a detour and figure out what the heck an antibody is. You know what an antibody is? Use them in lab. Very good. These are proteins. Ooh, that's bad. Proteins that can stick to antigens. So antibodies stick to antigens. All right. Which antigen do you think those antibodies will stick to? Take a guess. The original non-self antigen. It's always specific. So the B cells are going to make antibodies that will stick to that original antigen. Let me show you a picture of the antibodies and the sticking. This one here is. All right. Nope, not that one. Okay. All right, so here in the middle, those 3 little cells with the spikes, these are B cells. The orangey dot things, those are the antigens. Which of those B cells matches the antigen? Look at the center one. This one can match or bind to the original antigen, the non-self antigen. Does that make sense? So once that happens, I'm going to proliferate to form a clone. Look at the clones and tell me what you notice about them versus the original.
One looks exactly the same one does.
They're exactly the same, right? They're clones. So if this one can stick to the non-self, these can stick to the non-self. And they're going to make babies like this with plasma cells. Here are my plasma B cells. Those are the antibodies, little Ys. What do you notice about those little Ys?
They have the same antigens.
Do they look like those? If they look like those, what can they do?
Stick.
Stick to those. So again, it's that specific idea. I'm going to make antibodies that can stick to the same shape. So here's the killer question. Why bother? Why am I making something that doesn't kill anything but sticks to it?
You stop it from infecting any more cells.
How would that happen?
Because it's pulling them before it can get
Ah, yeah, all right. Let's look here. I'm going to take an antibody and stick it to the antigen. And you're going to see these things here. In English, what are those three boxes showing? Clumping. I heard clumping. Good. Rootnation. You did that in lab with your blood type, right? You added antibodies to your blood and it clumped up. Precipitation, neutralization. But no matter which of those three words you look at, the antigen is being stuck by the antibodies. So in the First World War, right, between the no man's land, they had a bunch of barbed wire. What was the purpose of the barbed wire?
Keep people from crossing.
Keep people from crossing. So if you cross, what would happen to you on the barbed wire?
Get snagged.
You get snagged. And what does the machine gunner do when you're sitting on the wire and can't move?
Shoots
He kills you anytime he wants, right? That's the idea. If you are getting stuck together, you can't move. And what do white blood cells do if you can't move and they can? They will eat you. So if I can stick to the antigen, I can eat the thing because it can't run away. Someone's going to hold you down and beat on you. That's the logic here. So as I stick to the antigens, the pathogen can't escape. And you're forever dead. So the purpose of antibodies is to stick to the antigens to stop them from moving, to reduce spread. So I'm going to stick to the antigens to stop the pathogen. I'm not killing the pathogen. I'm holding you down. We call this marked for death. Anyone remember that movie? That was great, yeah. That dates me, right? That was actually a good movie. Right. So what I'm doing is I'm flooding the body with these markers that are going to stick to the bad guys and hold them down. We call this flypaper, right? The logic that you're going to be stuck. So the antibodies are sticking to the original antigen, so now you can kill it with white blood cells. So the white blood cells can come along and eat you whenever they feel like it. You can't run away, hence the barbed wire. Right? Yes?
Do the white blood cells eat the antibodies?
Yes, they will, because they're protein. They will end up eating those too. Right? But we have a couple more steps to do. Before we do that, let's look at an animation of this process before we continue on. Is here.
The specific immune response is divided into two categories: humoral immunity and cell-mediated immunity. A humoral immune response consists of the activity of B cells and the antibodies they produce.
Okay, so cell-mediated was the T cell we just did. But humoral, another way to say B cell. All right, so that's the one we're doing just now. Right? We got that.
The process of B cell activation is called clonal selection. During this process, a B cell with a specific antigen receptor recognizes an antigen, and this cell proliferates to produce a multitude of cells, also called a clone of cells, with the same specificity, which differentiate into memory B cells and plasma cells. The antibodies produced by the plasma cells bind to antigens to produce the humoral response.
So I'm flooding my body with antibodies. They're going to stick to the bad guy.
The antibodies produced by plasma cells have several functions. Since each antibody has at least 2 antigen binding sites, antibodies can bind to more than one microbial cell, resulting in agglutination or clumping. This agglutination helps remove the microorganisms from the body. Antibodies can also neutralize toxins, viruses, and bacteria by binding to critical portions of the toxin or blocking proteins necessary for attachment to cells. Antibodies also act as opsonins for phagocytosis and activate the complement system.
So we just said in English, I'm going to stick you together so you can't move. I'm going to call more white blood cells, more troops to come over here and eat you. And then you're stuck, you can't move, you can't secrete, you can't get away, so you might as well just be dead, because you will be. That's the logic. So the antibodies, B cells aren't killing anything. They're simply secreting antibodies. The antibodies are holding the pathogen in place. All right? Make sense? That's the B cell system. I'm making antibodies to you. PCC says, thou shalt know all the antibody types thou shalt know. So guess what we're gonna do? Another big chart. What?
Never done one of those.
Yeah, so over here, make another big chart.
Is it